Today we will discuss about melanoma or skin cancer dysregulation. So what are the intercellular changes that occurs that might promote the tumor progression in the case of melanoma? Though dysregulation occurs in in almost all of the cancer cells. Here we will discuss about the dysregulation in terms of the glycosylation that occurs on the tumor, and that helps in the promotion of tumor either in terms of metastasis, migration, or uncontrolled growth rate. So the glycosylation in the melanoma that occurs when the melanoma is not in the cancer stage is shown on the left hand side. That is, it has the sialic acids, fucose, glycanac, polylacnac, glycan branching, gallinocytes, and O-glycosylation. However, when this melanoma becomes a tumor, or it has this, when it acquires the tumor properties, then this kind of glycosylation profile changes. For example, this P13-AKT pathway gets triggered because of the dysregulated glycosylation profile that occurs because of the high production of this FUT4 glycotransferase enzymes, and this will promote the cell survival even in the conditions where cells are not in a healthy state. And the upregulation of this GNT5 glycotransferase will cause this high glycanac related glycans, and that will promote FAK pathways. And similarly, this ST3 galvans are also ST3 galvan and galanT4 are also dysregulated. So in this case, what will happen is that the survival pathways gets upregulated, and that helps in this migration and invasion. So this kind of glycan structures helps in the migration and invasion also. However, this galanT4, this is dysregulated, and this helps in the survival of the tumor cells. That is, this kind of trial pathways, as we have discussed in our previous videos, will trigger apoptosis. So dysregulation of this kind of receptors will helps in reduction of this apoptosis pathways that could be triggered by these trial receptors. And other things that get upregulated is this high production is this dysregulation of this P38 pathways, or we can say that ERK12 pathways gets activated because of the high production of this glycanac structures on the O-glycan, or we can say TN or STN glycan structures. So this will inhibit P38 and will promote ERK12 pathways. Next, or going forward, now what will happen, this GCN2 is downregulated in the case of this melanoma skin cancer. So because of this, this IGF-1R signalings pathway will be reduced, and because of the reduction of this because of the reduction of this IGFR1 signaling integrin pathways, what will happen is that there is a production of this kind of glycosylation profiles without GCN2 will helps in the cell growth, because this can interact with the ECM and helps in this ECM composition changes that we have discussed in our previous videos. And the integrins also interact with the ECM, and within the integrins, this kind of integrins are upregulated. That is alpha salic acid alpha 2,3, this fucose alpha 2,3, fucose alpha 2, fucose alpha 3, and the dysregulations occurs in terms of the salic acid alpha 2,6 and polylacnac silatate is upregulated, and polylacnac fucose is upregulated. And this kind of integrins helps in this helps the cells towards migrations and also adhesion at the secondary sites. this is the impact of integrins. Lastly, things that are discussed is ICAM, L1CAM and MCAM, and because of the dysregulation of this 48 and GCN3 glycotransferase enzymes, so this will disrupt, or this will prevent the interaction directly to the receptors such as MCAM receptors or L1CAM receptors, because of the over glycosylations of this kind of receptors, and thereby this will prevent the cleavage of these enzymes and helps these proteins to stabilize, and this protein later on helps the cancer cells towards migrations or invasions. So let us just quickly revise about this IGF-1R signalings, as some people might have been in doubt what does these things does. So this GCN2 is downregulated, so it will not affect, or it will its function normally is to prevent this IGFR1 signalings. However, once it is downregulated, then IGFR1 signalings and integrin signalings could be triggered. That is, the resistance provided by GCN2 will be reduced, so that will cause the growth. Also, that will finally leads to the more production of this kind of receptors, IGF-1R receptors, and IGF-1R receptors thereafter helps in the growth of these tumor cells, melanoma tumor cells. This is about a brief overview of the dysregulation that occurs in the melanoma or skin cancer cells in terms of the post-translational modification, that is glycosylation. One of the post-translational modification, that is glycosylation. If you need to understand more about this dysregulations in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.